alaikum. Thank you so much for coming. I really appreciate that. First of all, I would like to thank organizer for giving us this opportunity to participate in such an amazing event about innovation. Also, I would like to thank all of you for coming to attend these sessions. The work which I am going to present here is a part of our ongoing projects, which micropropagations, phytochemical profiling and antioxidant activity of a pomegranate grown in Saudi Arabia. Before that, just would like to give you brief introduction about Istidama Center, the vision of Istidama, leadership in agricultural research and practices to contribute to food security and resilience. The visions enabling sustainable agriculture to enhance food security via applied research, development, and innovations. The center has 5 strategic objectives: creating knowledge in sustainable agriculture, promoting practices in sustainable agriculture, promoting innovation and adoption of modern technologies, building an efficient and effective institutions, establishing a stable and diverse funding base. So here, this is contents of our presentations. We have a brief introduction about pomegranate challenges, problem statement, justification, and objectives. Material and methods which is used in this study. We have results, future work, and conclusions. As you know, pomegranate is native to Central Asia and distributed among different climate conditions in tropical and subtropical regions. Also, pomegranate is among one of the most significant cultivated crops grown in tropical and subtropical regions. Besides, the nutritional uses of pomegranate has been used in traditional medicines, in folk medicines, to treat different type of disease, such as cancer prevention, antimicrobial, and diarrhea. There are different cultivars cultivated in Saudi Arabia, in different regions in Saudi Arabia of pomegranate. Among them, we can say Beta pomegranate has a unique sweet taste and high yields. Recently, there is a study used ISR reported that Beta pomegranate has unique genetically or may adapt to the location, be the locations. Also, MEWA has launched a program to expand cultivation of pomegranate. On the top of that, pomegranate has a cultural dimension, which is a festival celebrated annually according to its nutritional value and market demand. Problem statement: as you know, pomegranate plant is propagated by rooting of hardwood cuttings. However, establishing new plant, just seedling, it takes about 1 year just for seedling. Then the farmer should transplant it into soil. I think this is then the farmer should wait at least for 2 or 3 years to get fruit. Therefore, alternative method for accelerating to accelerate production is really needed. Here therefore alternative methods, as we propose here, should be micropropagation. Micropropagation techniques itself face some problem about phenolic compound. The plant contain high amount of phenolic compound, and it's not easy to propagate by micro techniques. The objective of this study, develop an efficient protocol for beta pomegranate productions, identification and estimation of bioactive compound of pomegranate, determination of antioxidant activity of beta pomegranate extract. Excipient, the excipient were collected from Al-Baha region. Then the cuttings were sterilized using our lab protocol. Then the cuttings were inoculated into different type of media. We used MS media, B5 media, N6 media, and woody plant media for plant growth. Genetic stability. For genetic stability, DNA was isolated from mother plant as well as from mother plant and from micropropagated plant. Then ISR-PCR techniques was performed. For bioactive compound extraction, here we use different methods for extraction of bioactive compound. We use succinate method to extract the bioactive compound, as well as we use our incubator to extract a bioactive compound from different plant extracts. For identification, the sample were submitted to different samples submitted to GC to see a bioactive compound in extracts. While for estimation, here we estimated different type of phenolic content. We estimate total phenolic compound. We estimate total flavonoid as well as total tannin content. For antioxidant activity, we use different techniques to estimate antioxidant activity of plant extract. We use ABTS, DBBH, and FIRAP. Here you can see different type of media we use. B5, MS media, woody plant, and N6. Those media supplemented with different BGRs concentration as well as with different antioxidant substrate such as citric acid. So we calculated the result based on survival rate of shoots, average numbers of shoot, leaf, roots, and callus weight. Here we can say the best media for shoot induction, callus induction was N6, which achieved best result according to these parameters. For shoot regeneration and multiplication, the same. We have MS media, woody plant, N6 gives same results as well as a root development. Actually, we used two models. Here, we use two models to propagate this plant. We use direct organogenesis, which here in these models, we use we induce shoots from excipient directly. Then the shoots were regenerated multiplied from induced shoots. Then the shoots rooted. On the other side, callus were induced from excipient, then regenerated shoot from callus, and root induced from regenerated shoots. This is the result of phytochemical analysis. What we did exactly, we injected different samples to see the phytochemical compound of our extract. So here we have different extract. We have shoot, callus extract. In shoot, we found about 25 bioactive compound, which has, I can say, which have strong biological activity against different disease. Also in callus, we get about 18 18 compounds. There is a variation between colors and shoots. Red colors indicate the high yield, while the green indicates lower yields. On the others, you can see here the phenolic compound. We detect total phenolic content in both shoot and colors, total flavonoids, and total tannins. Both colors and shoots contain high amount of phenolic compound. In compare, I think total phenol content highest in shoot compared to the colors, as well as total flavonoid content and total tannin content. This here we use ISR for genetic analysis to see is there genetic variation between mother plants and micropropagated plant. This is a pre-screening. We screened about 25 ISR primers. Out of them, 20 ISR primers resulted in 98 bands, and we can say here there is no genetic variation between mother plant as well as micropropagated plant. Also, we test, as I mentioned before, we test different antioxidant activity, use different methods. We use ABTS, DBBH, and FIRAB. And here we, as you see here, the inhibitions of free radicals increase with the increasing of extract up to 200, about 93 of free radicals, 93 of free radicals already scavenged by using this concentration. Based on this data, IC50 was calculated. Here, as you see, IC50 is lower. That means pomegranate has potential to be drug discovery. This future work, expanding research and application about the plant. We are going to also to study phenolic compound, study adaptation of the plant. Also, field study should be evaluated to see if there is a genetic variation between micropropagated and mother plant. Also, we are going to evaluate the nutritional value of pomegranate, potentiality of how we see for germplasm we need synthetic gene seeds for crop observations. Also, biological activity assessment, we are going to isolate different bioactive compound by using HbA1c. Then we will evaluate against cancer lines as well as against microorganisms. So here we conclude that this method has significant or creates significant advance to overcome the difficulties of plant reproduction, germplasm conservation, and gene editing or mass propagation materials we can provide. Also here we generate new source of bioactive compound by transforming shoot into colors. Based on ISR result, indirect organogenesis can be used for mass production. أود أن أتقدم بخالص الشكر والتقدير للدكتور خالد الرحيلي مدير مركز استدامة على الدعم لا محدود في المجال البحثي وبتابع معنا يوم بيوم. شكرا دكتور خالد. أيضا الدكتور قاسي السباعي رئيس قسم المختبرات بمركز استدامة. شكرا دكتور قاسي. عبد المنعم مدير مختبر زراعة الأنسجة النباتية والتقنية الحيوية. شكرا. عبد الله الحربي هو من عد المحاضرة. شكرا جزيلا عبد الله. Also I would like to thank شكرا جزيلا ندى، هي من قامت بكل العمل طبعا. just I am presenter here. هي من تسجل كل البيانات، بتعمل كل التجارب، وبالتالي الفضل يرجع لندى ما لي انا. شكرا منيره، برضه ساعدتنا كثير جدا جدا. Also I would like to thank my colleague from KSU, Dr. Nadim, Dr. Salim, Dr. Muhammad Tarroum. Thank you so much. Once again, thank you.
Thank you so much, Dr. Abdalrahman. Any questions from audience, please?
Hi. So,
Okay, welcome, Prof.
thank you.
You
Just a quick question with regard to pomegranate. Your work clearly makes it easier to propagate, but is there a significant market for pomegranate, or is there a likelihood that pomegranate will become a significant crop for Saudi Arabia as we go forwards?
Again, I didn't get you well. Just you can rise.
Is pomegranate going to become an important crop for the kingdom?
Yeah. According to this Mewa strategic is going to be.
Yeah, it is very important. It is very important because the species here, or the variety here, it's local from Al-Baha. They call it Beda, and this is one of the strategy to preserve the natural resource. And it is a very successful plant in Al-Baha, and there is a plan from Mewa to expand the plantation of pomegranate in Al-Baha.
So native species.
Yeah. Thank you.
Okay, thank you, Dr. Khalid.
Thank you.
Okay, we have a question in the
السلام عليكم.
عليكم السلام.
ما شاء الله عمل ممتاز، واعتقد زراعة الأنسجة في مجال النباتات يعني من المجالات اللي بتحتاج يعني شخص يكون يعني عنده خبرة وعنده اتليست بيرسونال هايجين وكده. فأنا بس عندي تعقيب على الانتي اوكسيدنت اكتيفيتيز، لانه يعني اذا كانت تارجت انتي اوكسيدنت اكتيفيتيز الفاينل برودكت ممكن يكون نيوتريسيوتيكالز برودكت او يعني مثلا سبلمنتس او كده. لكن لو حبيتوا مثلا تمشوا اكثر از انتي كانسر ولا تشوفوا از دراج ديسكفري، I think antioxidant is not enough, so you have to go like further to look for collaborators, or you can establish the research units that can do the drug discovery for you. So at this stage يعني في هذه المرحلة أنا بعتقد أنكم نجحت، يعني حققتوا نجاح كبير جدا جدا، ولكن المستقبل يعني ممتاز جدا، لأنه النجاح في عملية زراعة البومينجريد نفسها في المعمل هي يعني يعتبر من الإنجازات العظيمة يعني، لكن يجب أن يترجم في شكل مثلا supplements أو في شكل أدوية أو كده يعني. وأشكركم جدا جدا.
شكرا.
السؤال إنه يعني الخطة بتاعتكم شنو beyond the antioxidant activity
يعني احنا في البدايه رايحين تجاه الماس برودكشن اللي هو للنبات، دي اللي هو الاساس. اساس المشروع هو الاكسار الدقيق للرمان، ونحن ما اي رمان، اخترنا افضل صنف مميز بناء على المعلومات اللي عندنا او الداتا الموجوده. الوزاره هي اللي جمعت النبات، بالتالي اللي هي نحن ماشيين تجاه انه اكسار النبات بكميات كبيره عن طريق زراعه الانسجه. اذا انت عندك مثلا شجره واحده او شجرتين، عشان تكاثر او تطلع منها مثلا 25,000 تحتاج كم سنه؟ مثلا 10 15 سنه، وبالتالي اللي هي عن طريق هذه التقنيه نحن ممكن ننتجها في خلال سنه او سنتين او اقل. لكن في مشكله نحن واجهتنا، اللي هي النبات بيعمل مثلا اوكسيديشن بالفينول وكده. الفينولات كانت عاليه جدا، تقريبا بتمثل حوالي ممكن اقول 50 او 40% من الدراي ويت او وزن النبات. خلاص عشان نتعالج منها احنا رحنا مشينا عملنا انفستيجيشن، شفنا الكلص وجدنا الكلص فيه كميه اقل من الفينول. ما وقفنا هنا، رحنا نكاثره عن طريق الكلص. في نفس الوقت نحن عرفنا الفيتوكيميكالز السكندري ميتابولايتس دي كمرحله اولى. but we are going to validate it. حنعزل عندنا شباب مختصين في المجال، حنعزل المركبات هذه بالتاكيد مع عندنا تعاون مع كليات الصيدله، حنمشي اكثر في هذا المجال. اذا الاجابه كده واضحه ولا
لا؟ الاجابه واضحه، وانا حقيقه ما كان هو سؤال، انا بس جست يعني اشاده بعملكم.
شكرا
عمل طيب، وكونه تساعدوا المزارع او الدوله في انه يعني عمليه الاكسار بتاع الشجره نفسها، ده انجاز عظيم يعني.
شكرا.
ومأجورين باذن الله.
Any question for Dr. Abdalrahman? Okay. Thank you, Dr. Abdalrahman.
Thank you so much.